Good morning, everyone, and thank you for joining us today. I'm really excited to welcome you to our webinar on advancing respiratory drug development using translational mouse models. Respiratory disease remains one of the most complex therapeutic areas in drug development, and today we'll walk you through how carefully designed in vivo models can help bridge the gap between early discovery and meaningful clinical outcomes. My name is Mark Johnson, and I am GemPharmatech's Director of Business Development for the West Coast of North America, and I will be moderating. Today's scientific content will be presented by Dr. Yingshan Chung, who focuses on respiratory and infectious disease research and will guide you through the disease biology, our models, and some efficacy data. I'll start us off with a quick introduction to GemPharmatech and our approach, and then my colleague will dive deeper into the respiratory disease platforms and data. At GemPharmatech, our mission is simple but ambitious: to engineer a healthier world by pushing the boundaries of biomedical innovation. What that means in practice is providing not just mouse models, but integrated data, disease insight, and experimental design support to help teams make confident decisions earlier. Our strength comes from combining the world's largest library of mouse models with deep disease expertise, supporting everything from biomarker discovery and disease induction to pharmacology and efficacy testing. We see ourselves as an extension of our customers' research teams, especially in complex areas like respiratory biology, where model selection really matters. One thing that truly differentiates GemPharmatech is scale. We maintain a library of over 25,000 genetically engineered mouse models, and we continue to generate roughly 6,000 new strains each year, supporting more than 1,000 preclinical studies annually. This scale allows us to move quickly, whether that's customizing a model, reproducing published phenotypes, or supporting parallel programs across multiple disease areas. Operationally, we're a global organization with headquarters in Nanjing and San Diego, and multiple sites across Asia and the US, which allows us to support programs from discovery through IND enabling studies. With that overview, I'll hand things over to Dr. Chun, who will walk you through the respiratory disease landscape and the specific platforms we've developed. Take it away, Dr. Chun.
Thank you, Mark. Hello, everyone. In today's webinar, we are going to dive deep into the establishment, validation, and application of preclinical mouse model in the respiratory disease. The webinar contains three parts. First, we will make a brief introduction about the background of the lung disease, and then I will introduce you the platform for respiratory disease research. Last and most importantly, we will dive into the pulmonary disease mouse model and drug efficacy evaluations. First, to set the stage for our deep dive into the mouse models, let's first revisit the respiratory diseases that drive our attentions. Generally speaking, the respiratory diseases encompass a broad spectrum of disorders affecting the airway and the parenchymal of the lung, causing acute and chronic inflammation. Clinically, they are categorized into several high-priority groups. The first one is the chronic inflammatory disorders, which is also the airway diseases, like asthma and chronic obstructive pulmonary disease, COPD for short, categorized by the airway hyperresponsiveness, inflammation, and progressive lung function decline. The second one is the parenchymal lung diseases, like acute respiratory distress syndrome, the idiopathic pulmonary fibrosis, and other interstitial lung disease, marked by the abnormal tissue scarring that irreversibly impair the lung function. And these kind of diseases have high mortality rate and limited treatment options in clinic. The third part is the infectious respiratory diseases. The viral pneumonitis caused by influenza, respiratory syncytial virus or coronavirus infections, and bacterial pneumonia, which pose acute threat to the vulnerable populations. And next, to inform our choice and application of mouse models, it is critical to first clarify the structural differences between the marine and the human respiratory tract. As these distinctions directly impact the translational relevance for drug development and our choice of the preclinical trial. As you can see in the slide, in the left side of the slide, here is the structure of the respiratory tract of human and mice. We can see that humans have respiratory bronchioles, but mice don't. Their airways go straight from the terminal bronchioles to the alveoli. So physiologically, the mice rely fully on the alveoli for gas exchange. So airway irritation or blockage causes a faster oxygen drops in the mice. And next, let's see the right side of the image. Both mice and humans have similar key cell types in their airways. For example, the epithelial cells and the immune cells and the fibroblasts, but their numbers and distribution are totally different. Now, there is a key trend shaping the respiratory drug development and the preclinical evaluation needs. That is the growing focus on the inhaled formulations. With respect to the respiratory diseases, major pharmaceutical companies or the newly biotechs have poured substantial resource in recent years into the development of the inhaled formulations. This approach offers rapid onset, lower systematic exposure, and direct drug delivery to the lung. And alongside this shift, a wide range of nebulizer, as you can see in the slide here, and the inhaler have emerged. And this create a preclinical challenge meanwhile. That is how to model this uh, deliver route as accurate as possible. This means our preclinical models can no longer just assess a drug's biological activity alone. Instead, they must also mimic the real delivery scenarios. To help develop high-quality mouse model for the respiratory diseases and provide a reliable efficacy testing service, we need to build up a full and all-in-one system platform for this kind of research. First, as shown to ensure the stability of the respiratory disease modeling and the precision of the subsequent drug administration, we have major techniques including the nasal installation, tracheal installation, and tracheal intubation. Additionally, our platform is equipped with drug generators and different kind of exposure systems. As you can see, the mouse lung image on the slide demonstrated the distribution of the drug solution after the tracheal installation of the event sprue in mice. And in addition, for most respiratory diseases, the functional diagnosis is indispensable for both clinical diagnosis and therapeutic efficacy evaluation. And currently in clinic, the spirometry and whole body plethysmography chambers are commonly used in clinical practice, and correspondingly, our platform is also equipped with the instrument for mouse model testing. The non-invasive plethysmography chamber here allows for the monitoring of the changes in the tidal volumes and the respiratory rate in the mice during their quiet breathing. And when combined with the stimulant, it can also detect the alteration in airway responsiveness. On the other side, the invasive pulmonary function analyzer provide a more comprehensive assessment of the functional changes in the lower respiratory tract and the lung parenchymal of mice. For the establishment of the respiratory infectious disease model, Our platform is equipped with a certified biosafety level 2 facility, which is capable of undertaking the experiment involving a variety of pathogens. So behind our specialized in vivo model pipeline platform, our entire group also boasts a robust in vitro testing service platform, which enable accurate and standardized analysis of all samples generated during all the experiments. And supported by the above technical platforms, we have established a variety of respiratory disease models to meet the diverse need from different clients. Here you can get an overview of them at a glance. Now I'm going to show some typical lung disease models, including the background, pathogenesis, detailed model data, and application scenarios. First, let's take a look at the most typical disease related to the air sac, the acute respiratory distress syndrome, the ARDS, or acute lung injury. The ARDS refers to a life-threatening acute respiratory failure, which is triggered by various direct or indirect lung injury, such as the severe infection, trauma, and aspiration. It is marked by the impaired gas exchange function and reduced lung compliance, often requiring the patient often requiring mechanical ventilation support and posing a high risk of mortality in critically ill patients. The ARDS arise from the infectious or non-infectious insult that trigger a dysregulated lung inflammatory cascade with neutrophil as the key mediators. Initial stimuli activate the alveolar macrophages and which secrete chemokines to recruit the neutrophil to the lung. So the activated neutrophils release damaging enzymes, for example, the neutrophil elastase and caspase G that that degrade the alveolar capillary barrier structures. And excessive netosis further induce the cytotoxicity and the tissue destruction, causing the hypothermia and the respiratory failure. In preclinical, there are various methods for establishing the ARDS mouse models, and the most widely used approach currently is direct respiratory tract installation of LPS. LPS is an inducer to trigger the acute lung injury and the inflammatory response, and such reaction induced by this model is extremely rapid. As shown in the slide, we first examine the functional and the pathological changes in the mouse lung. Pulmonary function results indicate that after LPS induction, the overall functional residual capacity, the FRC of the lung, increased significantly due to the lung injury and edema. While the expiratory while the expiratory obstructive also increased slightly, the pathological findings demonstrate that even at 24-hour post-infection, post-induction of LPS, there was still obvious inflammatory cell infiltration in the lung tissue. And next, let's zoom in on the inflammatory response and enzymatic activity changes in the mice after the induction. At 4.5 hour post-induction, there was a significant increase in the neutrophils and the macrophages in the mouse bronchoalveolar lavage fluid, the BAF, and accompanied by the marked upregulation of TH1 type inflammatory cytokine expressions. We then enrich and collect these cells and detect the activation of their enzymatic activity. And the result shows that the core proteins, including the neutrophil elastase, the caspase-G, and PR3, were all activated. And these three enzymes collectively exacerbate the lung tissue damage and amplify the inflammation by degrading the alveolar capillary barrier and promoting the pro-inflammatory mediator release. The critical role of this enzyme activity makes them the key targets for the drug development, such as DPP1 inhibitors. The DPP1 inhibitors curb the activation of the proform of the enzymes like NE, caspase-G, and PR3 in the neutrophil granules, thus inhibiting the nature of protease activity. And we use the DPP1 inhibitor as our positive control drugs here. We analyze the neutrophil recruitment in both lung and bone marrow, and plus the enzyme activity of the cells in the above. The result clearly show that the acute lung inflammation triggered by the LPS was effectively kept in check by this DPP1 inhibitor. And in a nutshell, our LPS-induced ARDS model recapitulate key disease features and reliability validate the drug efficacy. Next, let's move on to the typical lung parenchymal disease model, the idiopathic pulmonary fibrosis. Idiopathic pulmonary fibrosis is a prototype of chronic, progressive, and fibrotic lung disease. The healthy tissue is replaced by the outer extracellular matrix, and the alveolar architecture is destroyed in the patient. IPF is a high mortality disease, and the ongoing injury disrupt the lung tissue repair balance, causing the abnormal crosstalk between the damaged epithelial cells and the fibroblast. This leads the fibroblast to become matrix producing myofibroblasts and driving the massive abnormal extracellular matrix buildup and pathological lung interstitial remodeling. And over time, the functional alveolar tissue is replaced, the gas exchange is impaired, and the respiratory function declines progressively. In preclinical study, bleomycin is commonly used to induce a pulmonary fibrosis phenotype in the mice. The bleomycin binds to the DNA and generate free radicals, causing the lung cell DNA damage, triggering the abnormal tissue repair and inflammation, and ultimately driving the lung fibroblast activations. For the model's establishment, we administer bleomycin installation on day 0, and we continuously monitor the mice body weight throughout the process and collect the lung tissue for pathological analysis on different time points, as you can see in the slide. And additionally, on the day 21, we also collect the BOF from some mice to detect the inflammatory factors, which help us to confirm the occurrence of the pulmonary inflammation and fibrosis. Now let's moving on to the next model. We will introduce the typical obstructive airway disease. And the COPD is characterized by the persistent airflow limitation that is not fully reversible with core pathological features, including airway inflammation, mucus hypersecretion, and alveolar destruction, often driven by long-term exposure to the harmful stimuli like cigarette smoke. The COPD exhibits significant clinic heterogeneity in its inflammatory phenotypes. Some patients predominantly present with a TH1 type inflammatory response, while others have evaluate eosinophil levels and manifests a TH2 type inflammatory profile. Although the number of the COPD patient is large, current treatment options are very limited. The clinical treatment is basically confined to three major classes of drugs, uh, like beta-2 agonists, the anticholinergics, and the inhaled corticosteroids. So, in response to these substantial unmet medical needs, the global pharmaceuticals companies are accelerating the research and development of the innovative drugs. In July 2024 and May 2025, Sanofi's dupilumab and GSK's mepolizumab were approved for the use in eosinophilic adult patients with COPD. And in the Jan 2024, the ensifentrine was approved. It is the first in over 30 years in health therapy for the maintenance treatment for adults with COPD. And these drugs combines bronchodilators and anti-inflammatory activities in one molecule. Given that it is difficult to recapitulate all the features of COPD with a single model during preclinical mouse model establishment, our platform has developed multiple models. The first one is a classic type 1 inflammatory model, primarily induced by the cigarette smoke. And the second one is the model with pulmonary type 2 inflammatory phenotype, co-induced by the cigarette smoke and the house dust mite. The third one is a pausing pancreatic elastase-induced model, with the emphysema as the main phenotype. First, let's take a look at the most classic model, the type 1 inflammatory model, primarily induced by the cigarette smoke. Generally speaking, this is a rather time-consuming model to establish. Mice are often required to undergo at least 3 to 4 months of cigarette smoke exposure via an nose-only exposure tower, and additionally to further stimulate the inflammatory response in the mice. A single dose of LPS is administered via instillation in each of the first 2 weeks of the protocol. And at the study endpoint, we collect the mice blood, both and lung tissue for respective axes, and conduct the lung function test simultaneously to better evaluate the model's phenotype. From the body weight changes, we observed that the mice exposed to the cigarette smoke maintains a stable body weight, while the control group mice show continuous weight gain. In terms of the physical appearance, the smoke-exposed mice exhibit a shaggy hair, irritability, and decreased activity. From the above, we detect the inflammatory cells and cytokines. The result shows a predominant increase in neutrophils along with the increased level of the cytokines such as IL-1 and IL-6. However, since the disease was in a state of in a chronic inflammation, so the expression level of these cytokines were less pronounced compared to those in acute inflammation models. And we focus simultaneously on the pulmonary pathological and the functional changes. From the pathologically, the HE stain revealed the emphysema phenotypes in the lung parenchymal, inducing the alveolar sac fusion and alveolar wall destruction. We also quantify the alveolar spectrum thickness and the average alveolar area of the objective assessment. In the bronchial region, the inflammatory cell infiltration was evident, and the muslin staining indicate collagen deposition around the airways. These indicators indicate that there is bronchitis happens in the mice. And now let's shift our focus on to the invasive lung function data, which directly mirror the obstructive and structural lung changes we observe in the pathology. First, given the presence of the emphysema in our smoke-induced model, we see clear increase in the total lung capacity and cord length. These evaluations are key indicators of the lung hyperinflation and alveolar wall destruction. In addition, we also detect a significant decrease in the expiratory volume, the FEV 0.1 and FVC ratio. This is a core functional marker that signal expiratory airway obstruction and defining the feature of COPD's airway limitation. Together, these lung function metrics align perfectly with the pathological findings previous. And as we mentioned that our cigarette smoke and LPS model replicate the classic TH1 type inflammatory phenotype. This type seen in roughly 60% of the patient and dominant by the neutrophil-driven inflammation. But in clinic, around 30 to 40% of the COPD patients have a TH2 dominant profile, marked by high eosinophil levels, and this group face a far higher high risk of acute exacerbation. To address the need for testing the treatment targeting this subgroup, we modify our existing model. We add the allergen host dust mite as an extra inducer, creating a model for type 2 inflammation, COPD. In this model, we collect the mouse samples for lung tissue RNA detection and both supernatant inflammatory factor protein analysis. The results show that after the induction of cigarette smoke and HCM, the lung exhibit a distinctly Th2 dominant inflammatory response. You can see in the slide here with significant increase in IL-4, IL-5, IL-6, and IL-13 levels. And further, for the pathological analysis of this model, we did more than just use the HE staining to assess the alveolar fusion. We also incorporate pus staining, and the result clearly showed a significant mucus deposition in the mouse bronchi after the HDM induction. This is a key feature of type 2 inflammation-related airway remodeling. And we move on to the respiratory function test. Here, the result is on the right side of the slide. We found that compared with the cigarette and LPS model, the HDM-induced model exhibit much more pronounced declines in the lung function, and particularly in terms of the airway obstruction, which align with the more severe clinical presentation seen in the type 2 COPD patient. Finally, let's talk about the PPE-induced COPD-like model. The PPE stands for pausing pancreatic elastase. When introduced into the lungs of mice, the PPE degrade the elastase fiber directly of the alveolar walls, causing pulmonary emphysema within just 3 to 4 weeks. Is pretty short. And as you can see from this experimental schematic, the PPE model is quite simple, but because it directly damaged the alveoli, its lung function changes are extremely robust. The static compliance, the lung volume, and the functional residue capacity all increase significantly. And while expiratory functions is reduced by roughly 50%. This makes it ideal for quickly evaluating the therapies targeting emphysema-related lung function impairment. And interestingly, uh, we also validate some findings from the published articles. That is, the PPE-induced COPD-like model is overall biased toward TH2 type inflammation. From the expression of inflammatory factors in both and QPs are result of related genes in the lung tissue, we can see that TH2 inflammatory factors in the lung increase significantly after the PPE inductions. And on pathological side, in addition to showing obvious emphysema, the pulse staining result also indicate significant mucus deposition in the airway of the mice. And these key findings making it a valuable tool for evaluating the therapies targeting TH2 dominant COPD. We've just walked through several classic non-infectious respiratory diseases and their corresponding mouse models. Here is a small conclusion, take-home message. You can take it as a note. And up to this point, for our final segment, we will shift to research on the respiratory infectious diseases. The respiratory system is easily invaded by different pathogens, causing local and or even invasive infection in our body. And our platform has built relevant models for viral, bacterial, and fungal lung infections. Here are two specific examples. And let's start with the first one, the respiratory infection with influenza virus. This model is commonly used to evaluate the protective efficacy of the anti-influenza drugs or vaccines. As illustrated here, we first vaccinate the wild-type mice and then challenge them with the influenza virus. We assess the vaccine's survival protection by monitoring the changes in the mouse body weight and overall viral survival rates. Meanwhile, we evaluate its viral replication and neutralization antibody titers and analysis the lung pathological changes. Actually, this experimental framework is broadly applicable to models of other kinds of respiratory infections, like the infection caused by respiratory syncytial virus or other kinds of bacteria. And next, it is worth noting that many human viruses are actually difficult to infect mice under the natural conditions. For instance, some highly receptor-dependent viruses will likely fail to complete the viral invasion in mice if the homology between the mouse and human receptor proteins is too low. To address this issue, we need to use the gene-edited mice where the relevant mouse protein is replaced with its human counterpart. And the example here demonstrates how humanizing the rhinovirus receptor ICAM1 in mice enabled a successful viral infection, overcoming the natural species barrier for this pathogen. We can see the viral replication and the infected mice causing pneumonia here. And here in the last slide, we showcase some of our available virus-related humanized or gene-edited mouse resource for the respiratory infection research. Actually, it also applicable for other infectious disease. For the humanization strategy, we offer both full gene humanization and extracellular domain-specific humanization options to meet different experimental needs. For any other target not listed here, please just feel free to reach out to us via the email for further consultation. Well, I believe that concludes my webinar presentation today. I hope the content share is helpful to you all, and thank you. And feel free to ask any question you may have, and you can type in the chat box or the Q&A section. Thank you.
Thank you, Dr. Chen. Thank you for staying awake so late to present this really cool data to us. Yeah, so if anyone has any questions, you can put them in the chat, and then I can read them to Dr. Chen and get you an answer. Okay, I have one first question here. It says lots of papers use whole body exposure setups for COPD models. Why
Mhm.
did you opt to use the nose only exposure tower instead?
Um, Thank you for a question. Actually, this is a very important issue even for us. Before the initiation of this project, we also have a deep discussion with these questions. The nose-only exposure can avoid smoke absorption through other body parts like the skin. Let's say if the mice were exposed to the smoke in a whole body chamber, it probably that many cigarette particles will stick in the hair or skin or other part of the body, and the mice will lick their hair, and so the cigarette smoke particle will get into the body by oral or skin absorption. That's what we don't want to see, because this absorption will cause the whole body inflammation. And the second point is that when we use the nose-only exposure, it can prevent uneven exposure caused by the mice huddling together, which means if the mice in the whole body chamber, they will huddle together when we expose the cigarette smoke to the mice. They will bury their head or mouth or nose into each other's body to avoid the smoke. So it can cause the variable effect in at last. So finally, we choose use the nose only exposure, although the uh, um, we although it is more expensive. Yes, thank you.
Thanks. I just learned something. Um, if anyone has any additional questions, please just send them to the email that we provided, sales@genpharmatech.com, and we can connect you to someone on my team, and also connect you to a scientific expert. So you can talk to Dr. Chen directly or someone on her team. We can also set up a follow-up call if needed. So thank you all for joining. Really appreciate your time. Hope you learned something. See you next time.